first international seminar series on liver toxicity and steatotic liver disease. I think I would like to give you a brief overview of this project. During the Pro-Euro-DILI network, the cost action, we had instaurated joint seminar conferences, but we wanted to continue this activity together with the coordinators of the European Whole Running project, Javier Cubero and Joana Miranda, who are the responsible of these educational activities, have thought of setting up a joint venture to create this international seminar series on liver toxicity and steatotic liver disease, which could serve to promote both projects. This is the first seminar, and in this very first seminar, we have the Professor Jonathan Fallowfield, whose research interests span basic science and translational clinical studies in hepatology. We are very much interested in the fields that he's developing and studying key topics, including mechanism of liver fibrogenesis and fibrosis regression, portal hypertension and hepatorenal syndrome biomarkers, including imaging. And for the whole running project, I think we also have the SteatoSITE web portal, which is going to be of great interest to all of us. So please, Jonathan, I give you the word, and thank you very much for presenting your experience and this project. Thank you.
Thank you. Thank you very much for inviting me. It was Lenny Nelson who invited me, who's our Edinburgh coordinator for the Holt Ronin Horizon Europe meeting. I'm aware there's a wider audience here, so I hope that this is relevant to everybody. So yeah, I'm going to talk about SteatoSITE today, and I'm based in Edinburgh. It doesn't quite look like this picture at the moment. It's very, very cold, and there's lots of snow in Scotland at the moment. And I'll just see if I can move my slide. So I'm based in a part of Edinburgh where the main hospital is centred, and it's co-located with most of the scientific buildings. So I'm actually based up here in the new Institute for Regeneration and Repair, but I'm nearby to medical informatics and health data science, the hospital, other scientific buildings. So for someone like myself that does translational liver research, it's a very, very good place to be. We've got access to patients and all the infrastructure that we need as well. So if anyone wants to come and visit or collaborate, then we've got good infrastructure. I've got some disclosures, but I don't think any of these are particularly relevant to what I'm talking about today. So I'm going to keep this quite straightforward, and I'm not going to go into massive detail when I'm talking about things. I'll let people ask me if they want to take a deeper dive into certain things. I'm going to very quickly summarize the clinical problem. Then I'm going to talk a bit about what SteatoSITE is and what we've done so far. And then I want to kind of open the door to what research I think we can use it for, and hopefully stimulate new avenues for collaboration. So as everyone is aware of now, there's been a big development in the last 6 months or so with the change in the nomenclature and the classification of what we used to call MASLD. And I think, you know, I've got mixed feelings about it, but I think in time it's actually going to be a helpful development. So what we used to understand as NAFLD in its purest form, we now refer to as MASLD. And then at the other end of the spectrum, of course, we have alcohol-related liver disease and various miscellaneous causes of steatotic liver disease. But then I guess the most helpful development from my perspective is there's now a recognized group with a proper name that is kind of at the intersection of metabolic-driven fatty liver disease and alcohol-driven fatty liver disease, and that's called MASLD. And I think that the thing we need to get used to as a liver community is to try and apply this new classification to cohorts we already have or cohorts that we are setting up. And that really does depend on a good alcohol history, which is not always available. So I don't want to tell people things that are too obvious, but there is this spectrum of MASLD going from a healthy liver through to a fatty liver, through to a fatty inflamed liver, and then a liver which accumulates scar tissue, eventually may become cirrhotic. And once cirrhotic, there's a higher risk of developing HCC. The curious thing about MASLD is that HCCs can develop in non-cirrhotic livers, which is something that we are still trying to understand. But you'll notice from the green figures along the bottom that this isn't a completely linear process, and there's a winnowing of numbers as disease progresses. So at least a third of the population now probably have steatosis, but then only a percentage of them will go on to develop MASH, and then even fewer will end up with cirrhosis and develop liver-related events. So just a very brief summary of MASLD. It's all, even in its previous guise as NAFLD, it's always been an integral component of what we know as the metabolic syndrome, although we've always recognized that at least 10% of patients have a non-obese phenotype. It's the main reason, certainly in the UK, I suspect everywhere else as well, in Europe, as the main reason for secondary care hepatology referrals. And prevalence is already high and is obviously on the rise. We do need to understand the pathophysiology better. We have a framework understanding, but we're always learning new things about this condition, particularly around the genetic and the environmental risk factors that are important here. Progression to cirrhosis and certainly liver-related events is extremely heterogeneous, hard to predict with the current tools that we have available, and in general is either slow or very unusual. So we need to identify subphenotypes within this condition to understand individual risk and to manage patients more effectively. That being said, cardiovascular disease is the leading cause of death in these patients. And I think this just accentuates the kind of multi-system and multidisciplinary implications of this condition. There is a clear lack of qualified biomarkers and disease-specific therapies, although, as everyone is aware, this is an area of intense activity. And because of the volume of disease and the fact that it is very difficult to identify on an individual basis someone's risk of progression, it represents not only a public health challenge, but also a precision medicine challenge. So, as I said, we don't really know which patients with MASLD are going to progress, but we need to, and I think this diagram here kind of highlights that. So if you take 100 patients with, this should be MASLD now, maybe only 5 will develop cirrhosis, and then only 2 or 3 of them will decompensate, and only 1 or 2 of those in total will develop a liver-related death or require a transplant over a prolonged period of time. So it really is imperative that we identify these high-risk patients, and even the opposite of that is also true. We need to identify the patients who are going to be okay, that we don't need to worry about quite so much. One thing that has become very clear over time is that liver fibrosis predicts outcomes. So this is data here that many people will be familiar with. This was the first prospective observational study in MASLD, 1,773 patients with a baseline biopsy, not that many with cirrhosis, and not a very long follow-up period of around 4 years. But what you can clearly see from the Kaplan-Meier survival curves here is that the more severe the fibrosis is, the more likely you are over time to either die or develop decompensation events. There are some questions about the representativeness of the patients in this cohort. I think what is fair to say is we need bigger studies with more diverse cohorts and longer follow-up. So is the reverse true? If we're able to reverse fibrosis, could that actually improve outcomes? And this is data from 2 negative drug trials that did not meet their primary histological endpoint. So simtuzumab and selonsertib in patients with compensated MASLD cirrhosis. And what this data showed in a post hoc analysis was that the patients whose fibrosis didn't regress had a much greater risk of liver-related events than patients who did show evidence of fibrosis regression. So it does seem to be the case that if you can improve fibrosis, you may well improve outcomes. But the NASH graveyard, as it's referred to, is all too familiar now, and there have been multiple pharmaceutical trials that have ended prematurely or ended in failure. And I guess the one shining light in all of this is the Madrigal drug resmetirom, which seems to be leading the way now in likely to be the first drug approved for this condition. But there have been many drugs that haven't haven't been successful, despite having quite promising preclinical and early clinical data. So it does make one wonder whether we're going about this the right way, whether our current model for drug development is the most effective, and what can we do to improve the chances of success. So we have a framework now, I think, a basic understanding of the pathogenesis of MASH that's founded on this sort of substrate overload hypothesis. But we really need to understand things at a much deeper level. And this is where multiomics is beginning to change the field considerably. And by learning about the genetics and the proteomics and the volatomics and the lipidomics, MASLD is slowly beginning to give up what may be its pivotal secrets to identify the most effective biomarkers and the most effective treatments. But the thing that I always wonder when I see these publications, some of which I've been responsible for or been part of, is, are these disease signatures a cause or are they a consequence of what we're seeing? And what are the prognostic associations? Because that really is important. So there are many cross-sectional studies where people have identified disease signatures, but it's really linking these to outcomes, hard outcomes, that is going to be most valuable. So I was really struck by this review article by Scott Friedman and Massimo Pinzani a couple of years ago, and this sentence I've highlighted here, which I totally agreed with at the time, and I agree with even more now, which is to really refine our treatment approach to MASLD and MASH. We need to take an approach that integrates genetics or omics, clinical data and pathological data in a very, very comprehensive way in order to identify the treatments that apply to specific subphenotypes of the disease. So this is a much more of a step towards precision medicine, which we really do not practice to any degree in hepatology, in stark comparison to oncology, for instance, where precision medicine is commonplace now. And I was really, really happy when I saw this paper and I read this sentence, because at the time we had just started to get to work on SteatoSITE. And SteatoSITE was a consortium, a small consortium by the standards of Holt-Ronin. It was funded by Innovate UK, which is the United Kingdom's innovation agency. So this is government funded. It was a precision medicine call that we applied to. And what we wanted to do was establish the first data commons for NAFLD, or now MASLD, research. And I was the clinical lead for this, and Tim Kendall, who's one of my liver pathology colleagues, was the pathology lead for this. We had a company called Eagle Genomics, a transcriptomics company, who were our kind of SME lead, and we collaborated with University of Glasgow, Precision Medicine Scotland, which is an innovation centre in Scotland, who hosts SteatoSITE, and other smaller groups within the University of Edinburgh. So what is a data commons? Well, it's an NIH term initially, and essentially it just means it's a platform, a database, if you like, to integrate data from diverse sources. And the idea around data commons were that they were supposed to break up these silos of data and integrate things like pathology, imaging, omics, and electronic health record data in such a way that they are accessible and easier to work with. And they were supposed to stimulate novel research that simply wasn't possible before, both from a sort of hypothesis generation point of view, but also for discovery and validation. So a little bit about Scotland, where I am, and its precision medicine ecosystem. So some good things first. We have a single unified health system in Scotland, and very highly developed infrastructure, both in the healthcare system and around health data science. We have a pretty stable, homogeneous population of around 5 million people. Every person in Scotland has a unique identifier that's used in every single contact that's made with the healthcare system, whether that's secondary care, primary care, anything, and that's called the Community Health Index number. And that makes it easy, in theory, to track any interactions people have with the health and care sector. There's a long-term experience with using electronic health records in research, and consequently some of the best health data in the world. And there is also a very high incidence of complex diseases in Scotland. I think it's only third in the world in terms of obesity, after Q8 in the USA. We have very high rates of obesity and cardiovascular disease, and MASLD. So you can see this table below. This is the European age standardized rate for chronic liver disease deaths, and Scotland is not only a lot further ahead than the rest of the UK, but also outstripping many other countries in Europe. So there's a high burden of liver disease, and there's a high death rate as well. So, right, I guess this is the key slide of my whole presentation. This is how SteatoSITE was constructed and what it looks like under the bonnet, I guess. So I'm going to talk you through this bit by bit. So I'm going to start down in the bottom left-hand corner here. So SteatoSITE was a tissue-first initiative. So we identified suitable cases through 3 of the 4 national biorepositories. And we identified almost 1,000 cases across the whole MASLD spectrum, which were, in the majority, were needle biopsies, but some of these were liver resections, some of them were liver explants for patients who had had a transplant. And these were all patients that were identified from the pathology standpoint, so with a histological diagnosis of MASLD. To try and reduce variability between labs and staining, we retrieved all of the paraffin blocks to Edinburgh and recut all of the sections from these liver samples and stained de novo for H&E and Picrocereus red, everything done in the same lab on the same machine in Edinburgh. Each stained section was digitized, and all the images have been curated. They were all scored histologically using the usual ordinal scoring scales, and also using continuous metrics like collagen proportionate area, fat percentage area as well. And as I'll touch upon later, we also had an unstained section which went for second harmonic generation 2-photon excitation microscopy. Then we took a thick curl from each block, and that was sent for RNA extraction. And I guess, surprisingly, considering some of these cases were quite old and they were all formalin fixed, paraffin embedded, we had successful RNA-seq data from 668 cases and 39 controls. And the controls were, again, identified from pathology. And these were cases where there was no evidence of any steatosis, any steatohepatitis, or any fibrosis in the sections. And we use these as controls for the RNA-seq. Then there was time-stamped clinical data from the electronic health records and other national data sets. So things like demographics, diagnostic and procedure codes. So for instance, if someone's had an endoscopy and had banding of varices, we get a code for that. Prescribing data, routine laboratory tests. So by this, I mean things that people would have if they come to the hepatology clinic or go to their GP. So we don't have access to something like ELF as part of routine care, but we can calculate things like FIB4 from routine laboratory tests. FibroScans, obviously, they've been around for quite a while, but maybe not forever. And the way that FibroScan data is recorded and stored from hospital to hospital seems to be quite variable. So although we collected as much FibroScan data as possible, I think that was one thing that we didn't have perfect coverage. And then obviously other key outcome data like death registry data. And then I guess the most important thing is, because every item of data, whether that be clinical data or the pathology or the sequencing data, everything has a time attached to it. So we used the date of tissue sampling as time 0, and that's this yellow vertical line. We went and collected clinical data from up to 10 years before through to up to 20 years following tissue sampling. So every single patient has an individual timeline which is decorated with a variable amount of clinical data, and it's different for everybody, but it's a very rich resource as a consequence. And everything has been integrated into one single database, SteatoSITE, which is hosted at the at Precision Medicine Scotland on their computing infrastructure. Now, I'm not going to pretend for a minute that this was easy. It wasn't easy at all. It was really difficult. This is big data, and it's a big data challenge. So in addition to all the RNA-seq files that you can imagine, and all the pathology images and all the pathology scoring, there are also multiple files of all the electronic health record extracts. So that comprised 38 CSV files containing 1,450 columns and over 1 million rows of clinical data. And that all had to be tidied up. And you can see here, I've just made a quick summary of each kind of stage or each element of the data where there were potential problems or issues. And you can see all the way through from the sort of simple things like spelling and spacing of data entry through to more sophisticated things like the way that data is coded or labeled, is all subject to potential errors and inconsistencies. So there was a very exacting data cleaning process. And one of the reasons for this is real world data. So we're dealing with something down here. We're not dealing with ideal data or things where there are simple errors. We're dealing with something that's very messy to begin with and needs to be tidied up. But after cleaning, restructuring, standardization by very, very patient data analysts, they resolved all the problems as far as possible, and we ended up with 534 distinct columns in the database, which is much more manageable. And in total, we had 5.67 million days of longitudinal clinical follow-up. So on an individual patient basis, this is roughly 7 years of follow-up was a sort of average. So a lot of clinical follow-up data. And the pain of doing this was recently rewarded when we finally published this. So this is the paper that we had published in November. And I think if you want to read in more detail, I suggest you go to the paper, because there's a lot more data and a lot more supplementary data that kind of fills in some of the gaps that I won't be able to fill in today. So what did the cohort look like in the end? Well, as I said, we had 940 patients we managed to identify. 659 needle biopsies, 225 resections, and 56 explants. 55% men, 45% women. The mean age was 55. The median BMI was 31, so they were obese. Around a third with type 2 diabetes. Mainly white Scottish ethnicity, and that's worth bearing in mind in terms of how generalizable or applicable the data is to other populations or ethnic groups. There are some caveats there, but all the deprivation indices were well represented. And as you know, MASLD is a disease of deprivation, and we see that very, very much writ large in Scotland, where there's huge variation in deprivation and burden of disease. This is just a way of summarizing the scored histopathological features. This is an alluvial diagram showing the relationship between steatosis, inflammation, ballooning, and fibrosis. And this is just 2 graphs here showing that as well as the ordinal scoring systems, we did do continuous analysis of collagen proportionate area and fat percentage area, and there was the expected relationship you would see with the ordinal scoring. Because everything was time-stamped and we were absolutely fastidious about that, that has enabled us to do time to event analysis. And this is a very outcome-rich cohort, which makes this kind of analysis very doable. So here, this is all-cause mortality. And I suppose that these first few graphs, this was one of the first analysis we did of the cohort to almost as a sanity check, because we were hoping, of course, that our Kaplan-Meier graphs would be broadly similar to the ones I showed you at the beginning in the Sanyal prospective study. And that is what we showed here. So there were 112 deaths from the biopsy cases. And what you can see here is the severity of fibrosis, the amount of scarring predicts the likelihood or risk of all-cause mortality. The similar pattern is shown here for hepatic decompensation. We used a composite outcome here using ICD-9 and 10 codes, and also OPCS4 procedure codes. So as I said before, like if someone had a large volume paracentesis or banding of varices, for instance, we used that as part of the composite outcome. And we identified a large number of decompensation events using this composite outcome in the biopsy cases. And again, this is a Kaplan-Meier plot. And we excluded cases where the decompensation event occurred before the biopsy. I mean, that was rare, but that's how you should do this analysis. And again, scarring predicts decompensation as well as it predicted mortality. And just to direct you, if anyone wants to do this type of research, then there are a few really, really important papers. And one of them is this one by Hannes Hagstrom and colleagues, where they kind of list the codes that you really need to make sure you capture if you're using electronic health records for research in this area. And again, I think, you know, the whole coding is going to change in the future with the new nomenclature. But suffice to say, these kind of papers are absolutely, you know, they're a lifesaver when you're trying to make sure you don't miss codes. Then the final thing, this isn't as pretty a graph. This is HCC development, and that's because there weren't as many HCCs. We did identify 80 cases where HCC was coded for in the electronic health record, but just bear in mind that some of these were explants, some of these were liver resection cases, and some were biopsies. So we only examined here a plot of the biopsies where the HCC coding occurred after the biopsy. Okay, again, so we're looking at the relationship here between scarring and HCC development, and you can see again a similar pattern, but although not as pretty because less cases. But again, very consistent as before. Then just a quick run through of some of the RNA-seq data. So we compared the controls and the cases. Here you can see on the left here, principal component analysis showing some segregation, moderate segregation according to fibrosis stage. And here you can see a kind of overall heat map comparing the very mildest end of fibrosis, F0, F1, compared to F4. And you can see here just at the overall heat map. One thing that we were always planning to do, although it didn't go in our original iteration of the manuscript, but we were asked to do by the reviewers, which was a very, very good exercise, was actually to stratify or disaggregate the gene expression data according to gender. And you can see here that the numbers of males and females in at each stage. And there were some really interesting things. I haven't got time to talk in great detail about it today, but one thing that's obvious here when you look at the differentially expressed genes is that in the very early stages of disease, there are far more differentially expressed genes in women compared to men. But then as disease progresses, this tends to even out. And there are a lot more genes, differentially expressed genes in common in the more advanced stages. This is a heat map looking at the top 20 most differentially expressed genes in each fibrosis stage in males and females. And one thing you can see looking at this kind of this way of summarizing the data is they're not identical. It's not a mirror image at all. There are some clear differences here. And that became evident when we did some pathway analysis. So first of all, starting on the left, this is not disaggregated data. This is the whole cohort data pathway analysis. Again, sorts of things you'd expect, enrichment of pathways related to extracellular matrix and extracellular matrix receptor expression as fibrosis becomes more severe. Interestingly, because of the link with cardiovascular disease, we see enrichment of pathways associated with cardiomyopathy. But then I guess the thing that was really striking for me was when you looked at the reactome data separated between males and females, you simply see some pathways that weren't enriched at all in females compared to males, or you see some differences, like for instance in collagen here. This is something that we have to look at in far more detail, but it clearly, aligns to what everyone has understood for a long time, which is that MASLD is a sexually dimorphic condition, and that there are some very, very interesting differences between males and females. And I think this is very, very pertinent when we're thinking about developing biomarkers and treatments. This is not necessarily a one-size-fits-all process, and maybe we need to think carefully about which drugs, which biomarkers are going to be most suitable for most people. One thing we were keen to do, just to sort of generate interest in the community and give people a taster, was to summarize the RNA-seq data and make it accessible by a web browser. So we created an R-Shiny app here, and this is just I've just taken a screenshot here of collagen 1, just so people can see what's really in SteatoSITE a bit more and generate some interest. So that's accessible to anyone and everyone at any time. Then we really wanted to start using this resource to identify, I guess, genes and other factors, tissue factors that may predict the risk of future liver-related and non-liver-related events. So what I'm showing here is a transcriptional risk score that we created for hepatic decompensation. So again, I haven't got a huge amount of time to go into the methodology, but what we used here was a lasso regularized Cox regression method to reduce the number of genes. And then what we've shown here is, in the end, what was a 15-gene transcriptional risk score that predicted decompensation. Here you can see, you can stratify the whole cohort into high or low risk. And there's some interesting genes that came out in this 15-gene panel. So one of them, cordin-like 2, is known to be upregulated in tumor tissues, including HCC. Then others like staniocalcin 1 and glial cell-derived neurotrophic factor, staniocalcin 1, fibroblast growth factor 7, connective tissue growth factor. These have all been linked to hepatic fibrogenesis. And I think obviously this is tissue that we've used here, but at least 5 of these genes are predicted to generate circulating protein biomarkers. And certainly this is something that we would very much want to validate in independent cohorts. And if anyone has cohorts where you have serum and you have gene expression data and it's linked to outcomes, then maybe you're the people for us to work with to validate this. The other thing that we were keen to do was to go upstream and try and identify core gene networks or master regulators that were likely to influence disease progression and patient outcome. So we used the high-risk genes from the TRS to try and uncover master regulators. And intriguingly, there was one regulon, thyroid hormone receptor beta, here you can see it up here, which was suppressive of the other two that we identified, AEBP1 and BNC2, which are both linked to fibrosis and MASLD. And what we showed here is that the THRB regulon activity not only decreased with advancing fibrosis stage, but it also predicted the risk of hepatic decompensation. And it did this regardless of the fibrosis stage. So I showed here biopsy cases that were F0 or F1. And if they all got the same, they all start at the same fibrosis stage, but you can use the THRB regulon activity to split them into high and low risk of decompensation. And exactly the same is true for F4 fibrosis as well. And obviously this was, really nice data to see, and quite, pertinent, given that liver targeted thyromimetics such as resmetirom are kind of prime time in MASLD drug development at the moment. They, increase hepatic fat metabolism, reduce lipotoxicity. So this was really reassuring, and of course, people interested in this area were very reassured. So I think now I want to just quickly talk about how this resource can be used further for research. Obviously, some of these things we're doing ourselves already, but I think would be useful to other people as well. I think this is useful as an independent validation set if you've got genes or markers that or gene signatures that you have generated yourself that you want to validate, and an independent data set that's useful for that. I'll show an example of that, what we've already done with collaborators. Very useful once you get into the bioinformatics to use AI and machine learning approaches. One thing where you have electronic health record data at scale is you can start to apply the data to generate clinical decision support tools. Digital pathology, I think this is a very, very valuable resource for that. We have digitized images for all of these cases, and of course they're linked to outcomes. I think it's a good resource to develop new prognostic biomarkers, and equally to identify new drug targets and potentially drug combinations at different stages of disease, because everything is very well phenotyped across the MASLD spectrum. So very quickly, just exemplifying how the SteatoSITE can be used. This was a collaboration with an Australian group who were interested in HCC. As you know, HCC is a low incidence complication of MASLD. But the curious thing about it is it can occur in non-cirrhotic livers. We know some of the metabolic risks that make HCC more likely, but people are very, very keen to understand what happens at a molecular and cellular level to drive HCC risk in MASLD. So this group were very, very interested in using single nucleus RNA-seq, and what they wanted to do was identify a pre-malignant cell type or cell state that connoted increased future risk of HCC development. So they had lots of mouse single cell data, they had lots of human single cell data, and they used SteatoSITE to help validate what they'd shown. And they identified this disease-associated hepatocyte, so-called DARHEP, which was consistent through different data sets, including SteatoSITE, and that was published last year. One thing that we have been doing more recently, working with this company here, Bering, who are an AI company, is to use the electronic health record data in particular to predict outcomes. And this is a schematic diagram here. This work was funded again by Innovate UK and Enterprise Singapore. And what we wanted to do here was to use routinely collected electronic health record data in SteatoSITE collected over a 10-year observation window to train a bespoke AI algorithm to accurately identify MASLD patients who are at increased risk of mortality over a 3-year window. So this is a bit more information about the model. This was a deep learning model. It was trained using something called 5-fold cross-validation that prevents overfitting of the model, whilst trying to take into account, make full advantage, if you like, of what in AI terms is a relatively small data set. And this schematic here at the bottom shows what the deep learning model consisted of, and it's something called bidirectional long short-term memory, and that is interleaved with something called an attention mechanism. And this deep learning model processes the input sequences in both forward and backward directions, so you capture information from past and future time steps, and the attention layer helps the model focus on the most relevant parts of the input sequence. So this data, this model that we generated here, we generated data and we validated it externally in an independent data set, and we're just writing up the data for publication at present. I can't really talk more about it, but it seems to be quite effective at predicting mortality. And again, it has been validated. So watch this space. Hopefully this will be publishing this data quite soon, I hope. And then the other thing I just wanted to touch upon was digital pathology. So another way that we can use the pathology here is to apply novel AI digital pathology methods. And what we don't want to do is what we already do using conventional pathology and do it worse. So the great thing about this is that we're using a different methodology that actually looks for features in the tissue that are completely inapparent to human pathologists. So SHG microscopy identifies the collagen, and the 2-photon identifies hepatocytes, and you get these beautiful pictures here. And what we did was all the biopsy sections from SteatoSITE we used to look for novel fibrosis-associated features. What had never been possible before was to directly predict outcomes from the tissue. We always have been using fibrosis stage as a surrogate, but SteatoSITE's given us the opportunity to do this. So what we did is we took the majority of the biopsies and we split them randomly into a training and an internal validation set. And we used the histoindex technology to image all of the sections. Then we used sequential feature selection, and 10 and 5 parameters were chosen out of a total of 184, believe it or not, fibrosis-related parameters. And we used linear regression to construct individual indices that predict all-cause mortality, decompensation, and HCC. And we did time-to-event analysis using Kaplan-Meier method, and we used death as a competing risk for decompensation, HCC, as you would. And then we compared distributions using the log rank test. And then we generated hazard ratios. This data we just submitted yesterday for EASL, so I don't really want to present the data now. Suffice to say that these novel indices that we generated massively outperform ordinal scores such as the NASH CRN and the Q fibrosis ordinal stage that HistoIndex generated. And then the very last thing that I just want to touch upon is how you can use SteatoSITE to identify novel therapies. And this is work that PhD student Maria has done in the lab using SteatoSITE, using computational bioinformatic analysis, and then in parallel developing an 3D in vitro iPSC trisphere model to test identified compounds with a major focus on repurposed compounds. And this is just another thing you can use SteatoSITE for is if you develop a model like these spheres here, you can then look to see how well your model correlates with something like SteatoSITE. And you can see here, interestingly, the spheroid model at maximum kind of activity quite nicely mimics the F4 patients in SteatoSITE, and key genes like aldoketoreductase reductase 1b10, which is known to be important in patients with NASH and fibrosis, is represented here. So I just want to wrap things up now. Sorry, we started a little bit late. We created this merged data set from multiple data providers across Scotland. So 12 of the 14 territorial health boards in Scotland provided data for this project, but final delivery was difficult. This was challenging. I'm not pretending it wasn't for a minute, but I think we do have now a really useful resource to support translational research. And initially we focused on using AI methods to look at the clinical data, to look at the pathology data, but there are loads more things we could do, and we're really, really keen to collaborate, not just with people who are within Ronin, but also people who may be on this call who are from different centres or different groups. So I'm going to finish there and just acknowledge multiple funders for the previous and ongoing work. Tim, who's my co-pilot on this project, and Maria, who's been just literally the best PhD student you could ever have. So I'm going to stop there and I'll stop sharing, and hopefully there's time for a few questions.
Thank you, Jonathan, for this excellent overview of the SteatoSITE platform. I think it's amazing the work you have done. And I would like to give the word to Javier Cubero, who has joined after he finished his class. You can take all the questions if you want. I will make a question later on. And Daly has raised her hand.
Yes. Well, just to thank you, Jonathan, before I pass the word to Anne. Thank you for giving us this amazing talk. We already read your paper, so it was really, really nice. And you have to tell us how you analyze this so many data and put it together with a real nice output, no, that you can really use in the clinical outcome. So, well, I mean, this is one of the things that because we as scientists, we get so many data, but we don't know how to interpret them. And in the paper, you see them so clearly. I mean, the outcome is so clear, and then you just test it in patients and you see that it's working. So you need to have really a good bioinformatician, right?
Yeah. I think the data is only as good as the people that and afterwards. So yeah, Maria, the PhD student I mentioned, she taught herself bioinformatics and she's been brilliant. But I think that will unlock further power and more secrets in the data by applying really a sophisticated bioinformatics method. So yeah, you absolutely need, if I was a basic scientist now, not knowing really what to do, I'd make sure I knew bioinformatics, because everyone's developing data now, but we don't know how to analyze it properly or apply it. So yeah, exactly. It'd be a really good thing to do to be good at bioinformatics. You'd be everyone's friend.
Yeah, I agree. I completely agree. Okay, Anne, do you have your hand raised?
Yeah. Okay.
Go ahead, please.
Hear me? Yeah.
Yeah.
Okay. This was a very interesting talk, Jonathan. Congratulations on your project and your nice paper. So I've got a quick question about you show a sex difference in your transcriptomics, which is very nice, something that we haven't been able to show in our similar study in some ways, probably because we're not adequately powered, since yours has many more cases in it. But have you looked at the effect of genotype on transcriptomics? Have you, for example, pulled out your PNPLA3 genotypes that are in your RNA-seq and looked at the sort of profiles you see comparing carriers and non-carriers? You could do this for other genes like TM6SF2 as well, but I think it's more easily done for PNPLA3.
Yeah, thanks, Anne. Nice to hear from you. So there's some things that we still haven't even, very basic things we haven't even done with the sex stratified data. We haven't actually divided people into if we could, premenopausal, postmenopausal,
Yeah.
or looked at the influence of HRT or anything like that within the prescribing data. So we need to do far more, I think, to tease out what a true difference is. The genotyping is a slightly I feel like I'm slightly on thin ice, especially speaking to someone like you about this. So this was bulk RNA-seq. So we did variant calling from the RNA-seq data with all the caveats that entails. PNPLA3 actually did look like we had quite good coverage, but we have not really, other than looking whether on its own it was strongly linked to outcome, which it didn't appear to be independently. We haven't actually looked very, very carefully at the variant calling data. It's one of the things that we want to do, probably with people that understand it better than us, who may actually go back to the original files and potentially use different methodology to generate nicer data. But we haven't, Anne, to be honest, looked at the influence of PNPLA3 or layered it onto any of the other analysis we've done. That's one thing that we need to do more with.
Yeah, well, your data is well placed, I think, to do this because you've got the adequate numbers. And I suspect that PNPLA3 is a good gene because of the fact that the key SNP is a coding region SNP, and it's not difficult to find in your RNA-seq. That won't be true for all NAFLD genetic risk factors, I suspect. But for the key one, it is.
Yeah, thank you.
Thanks.
Oh, go ahead.
Yeah, I have a talk, but I couldn't find the way to raise my hand. So, well, Jonathan, thank you for your talk. It was very interesting, and for the study. I mean, it's amazing work. So, you know that in running, we are developing a number of in vitro models that theoretically should try to mimic steatotic liver disease. How can we integrate our data with yours and to compare and to use and to put everything together? How would be the approach?
Yeah, so we, I mean, you can just, I mean, the simplest way is if you have gene expression data from your in vitro model, and then we just do a comparative analysis and see which genes are represented in SteatoSITE. The first initial thing to do. We had nanostring data actually from the in vitro model that we did, and that's what we were looking at. That graph I showed very quickly at the end was just a pretty rudimentary comparison between the two, just to look for overlap. But there are more sophisticated analyses you can do. But if you have a list of genes, we have a list of genes, we can look for representativeness of the in vitro model. I think that's an obvious thing to do and simple enough. We've done that with other collaborators and things as well. So yeah.
Okay, good. And well, you mentioned about the MASLD and how alcohol should be taken into consideration. How are you really planning to address that in your cohort, or is there any way of knowing if they were taking alcohol or not, or things like that?
So retrospectively, it's really, really difficult. As you can imagine, the information that you gather is only as good as the information that's been recorded by the clinician and provided by the patient. Over the last 10, 15 years, we'd never even contemplated anything like PEth testing or more objective ways of quantifying drinking, or certainly unanticipated drinking. And we know from studies, even in prospective studies, when you actually test in the hair for alcohol, maybe a quarter of people who say they're not drinking are drinking. So even in prospective studies, we can't be sure. So I think it's always a caveat. We tried as many simple ways as we could to identify alcohol that had crept in, but undoubtedly there's undoubtedly going to be some MASLD in our cohort. I'm sure of that, because we have no 100% bomb-proof way of knowing whether people, particularly retrospectively, I think it's very, very difficult. So you're relying on, you know, historical data, and we know that clinicians are not very good at recording alcohol. One thing we're trying to do at the moment is access primary care data on every patient to go back to the primary care records, because someone's family practitioner may have a better handle on alcohol consumption than secondary care physicians.
Yeah,
just maybe.
Yeah. Well, thank you very much.
Thanks.
Paula, go ahead.
Hi. Fantastic presentation. It's an amazing database. Congratulations. I have a question. Maybe you mentioned it in the presentation, sorry. Do you have serum or plasma sample from this patient for future analysis? Thank you.
I wish we did, but we don't. Unfortunately, in the hospital here and in most hospitals in Scotland, after a week, they throw away the sample, because this was all routine blood data we've recorded. And because this wasn't a prospective research study, we didn't have informed consent to collect blood. So we don't have any stored plasma or serum on these patients, which is the one thing that we're missing. And it's the reason why, if we want to validate things in serum, we need to find analogous data sets that we can use to fill in those gaps. So unfortunately, we don't. We only have the real, if you like, the real world laboratory data that's been collected over the last 20 years or so. Yeah, so we don't, unfortunately. I wish we did, but we have quite a lot of other things, but we don't have that.
Okay, thank you.
Maribel, you want to talk?
Yeah, well, I wanted to commend you and all the team for this huge effort to put together this wonderful, and to have the idea, no, and the vision to create this platform. And I would like if you could tell us, what do you think will be the limitations of this SteatoSITE website in terms of validation? Because I've seen that your cohort, how that could compare. For example, if we want to see, we find some interesting data that how could compare into your data set.
Yeah,
what do you think the limitations will be?
Yeah, so I think we have to be cognizant of the fact that the majority of patients in this cohort are white Scottish. So, is that necessarily going to generalize to, say, a Hispanic cohort? You know, quite possibly not. Some things may, but when you're talking about things like, you know, the genetics, for instance, then that may well not be suitable. But there may well be other elements that actually are not affected by the demography of the cohort, but some things likely are. And so I think, you know, the other thing is because this is a secondary care cohort, these are all patients that had some reason or another for a biopsy or an operation or a transplant. So there's inherent spectrum bias. So we can't generalize that that data for, you know, a kind of general population, natural history, for instance. This is a secondary care cohort. So I think there's inherent spectrum bias, and there's also, you know, there's quite, you know, I guess it's a single country study. So in terms of the demographics, we can't completely generalize to other geographic or ethnic populations. So I think those are important caveats, but they're more or less important for different types of research question, I'd imagine. Yeah.
And just some details. Do you collect medications that the patient had been been taken or have been owned?
Yeah, so initially, because what we've published and presented, I would call SteatoSITE version 1, but I'd like to have a version 2 in a year or so, where we have primary care data on top, and we have some coding data that we wished we'd collected the first time but didn't, so a few blood tests that we didn't ask for that we want to get, and also the full prescribing data. The first iteration of this, we were very focused. We wanted all of the diabetes medications, all the cardiovascular medications, statins, etc., and non-selective beta blockers, rifaximin, that kind of thing. However, someone came up to me the other day and said, "Oh, I'm interested in the effect of antipsychotics on liver injury and fatty liver or something. Have you got that data?" And I said, "We haven't got that data, but we should have it." So we need to fill in the gaps to have the full inventory of prescribing data. We at the minute we're relatively focused on, I guess, the most focused MASLD related drugs that people might be on, but we haven't got every medicine.
Just last comment, because I am interested to know, if once you establish a collaboration, the results that have been published, do you include them back into the data set?
That's a really, really good point. So that was always our intention. So, we want to collaborate with people, and we want to try and build this and make it stronger. So, for instance, the histoindex data, the stain-free imaging, all of the metrics from that study, that separately funded study, is about to be imported into SteatoSITE. So we have an extra data set on the pathology. Equally, if we generate new analysis from other projects, the intention is we would, if possible, again, build that into the data set. So we keep making it better and stronger, I hope.
Yes.
Obviously, if we collaborate with some industry, some companies, they may want to retain their IP, and that's fine. But most academic collaborations, we would be hopeful, once they've been published and everything's been sort of locked down, we'd be able to hopefully make that data available within SteatoSITE.
Thank you.
Yeah. Okay, Jonathan, thank you very much for giving us this first talk of the Young Investigator Seminars. And thank you everyone for your attendance. Remember that in February we have another talk. So we will send you the link to connect. So from my side, that's all. Thank you, Maribel, also for being there.
Thank you. Well, it will be interesting to point out that Anne Daly and Marina Villanueva are the MASLD Healing Consortium coordinators of the Educational Activities Committee. Also, who include also Anna Licata, Jane Grove, Liliana, well, from other countries. So next time they should take the word and make the presentations.
Okay.
Thank you, Maribel.
Thank you.
Thank you.
Excellent.
Thank you for having me again, and I look forward to seeing some of you in Edinburgh soon in the Ronin project.
Yes.
Yes.
We shall meet there in a couple of weeks.
Great. Thanks, everyone. Have a good day.
See you soon.
Bye.
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 Bye-bye.
Bye.